microscope. So I'm going to go through what you have missed quite quickly, but not very quickly, and then we shall start with a new topic of this week and next week, اللي هو الابيثيليوم. You have 2 lectures recorded للابيثيليوم اللي هم الجنكشن complexes. في كمان برضو another topic اللي هم ذكروني غير junction complexes شو فيه؟ للي فتحوا طبعا.
الابيثيليال apical.
Yes, exactly. الapical structures اللي هم السيليا, microvilli, and the villi. So next week we shall see real images of the epithelium. Now let's just get things clear here in the lecture. No smiling, unnecessary smiling is not required. No chatting during the lecture. I will have to embarrass you, either to relocate you or actually escort you outside. So please have the fullest respect to the lecture. Got the point? Got it? بالعربي. جنب زميلك ما بتحكي معه وما في داعي للابتسامات الغير مبررة. راح اضطر احرجكم واغير مكانكم، او اذا كان الاتيتيود يعني غير مقبول بتاتا راح اضطر اطلعك برا. تمام؟ Okay, thank you. So, human histology. What is human histology? ببساطة, what you have seen in the anatomy classes and practical, this exact same thing. You're going to look at it under the microscope. So ببساطة, those are sections of different tissues under the microscope. Do we need to know? Yes, we need to know. We need to know what are the type of tissues present in the wall of the stomach. We need to know what type of tissues are present in the skin. And this is what we will study. Not organs, not systems, لكن 4 basic tissue types. What are these 4 basic tissue types are called? Epithelium, the connective tissue. Now, 4 and 5 are actually subtypes of the connective tissue. So you can group them into one under the big name connective tissue. And then the muscle and the nervous tissue. So these 4, what we will discuss, we will see different images of all the types of the epithelium, of the connective tissue, and the types of the muscle, and the different types of neurons, human nerve cells, that we have in our bodies. Now, next year, this is not what you will be studying. You will be studying systems. راح تدرس respiratory tract, راح تدرس GI tract. فبتاخدهم. You already know how the epithelium look like. فلما بتاخد section بالstomach تعرف بالضبط وين راح تتوقع انه يكون موجود الابثيليوم. And accordingly, you should be able to identify the different types of epithelium present in different types of organs. تمام? So, level of organization. What does that mean? How are we structured? إحنا as human beings, what are we made of? ببساطة, we are made of systems. But these systems are simply composed of organs. ناخذ مثال. That's you when you were a little child. This is your GI tract. Gastrointestinal tract. نختصرها GIT أو digestive tract. So, in the digestive tract, in the respiratory tract, what do we have? We have a number of organs that they perform similar functions. GI tract شو وظيفته? Ingestion of food, swallowing of food, digestion of food, absorption of the end products, and then excretion. هاي وظيفتهم. فكل جزء من ال GI tract will be part of these processes. Now, if we take one of those organs, and that's what? That's the stomach. What is the stomach made of? You would think. In the back of your mind, you might think it's only made of one type of cells, one type of tissue, but that is incorrect. Then we have many functions that they are happening, or many events that are happening in the stomach that requires different types of tissues. يعني هل هاي الستومك عبارة عن ايش؟ عبارة عن empty sac, كيس فاضي. But this sac is able to what? To contract and relax. Correct or incorrect? So what type of tissue you would expect to find in the stomach if it can contract? Muscles, exactly. At the same time, this muscle, اللايننج تبعها is capable of secretion. عنده القدرة على انه يفرز. So شو بيلزموا معناته؟
epithelium.
These glands, that's what you would think and that's correct. مين التيشو اللي بيعطينا هاي الخاصية للسكريشن؟ Epithelium, and that's exactly what we have on the inside of the stomach. We have epithelium. So generally speaking, you would expect different types of tissues. There isn't an organ that is made of one tissue only. فكرة إنه many, because we anticipate different functions, and those will do these separate functions because they are different in their properties. So which one shall we take to expand? We will take the muscle layer of the stomach, and that's how it looks like. That's the muscle layer. What type of muscles we have in the stomach? اللي هم involuntary يعني لا إراديات. قديش في عندنا أنواع من المسلز لا إراديات؟ Two types, the smooth and the cardiac. والvoluntary biceps, triceps, yes. إذا skeletal هم الوحيدين الvoluntary, and this is a member of the involuntary. Those are the smooth muscles, and that's how actually they would look like under the microscope. This is a diagram of a representative diagram of the muscles. So if I take one of them, that's how it looks like. It's a spindle shape with the nucleus occupying the biggest part of the cell. Now, these cells, what are they made of? ببساطة من إيش معمولين الخلايا؟ من molecules, proteins, lipids, and carbs. Different types of them. Now, those different ones will be part of the cell wall, of the cell organelles, or the cytoplasm. Now, the next question is, would you expect all tissue to look the same? No, they don't. They don't, and that's how you will be able to identify them under the microscope, the light to be more specific. We're not going to be seeing many of the electron microscopes, but you will be exposed to it. So a smooth muscle will look differently, or different from a skeletal, different from a cardiac. A neuron is trillion% different from any other cell, and that's how we can distinguish them. Now, what is this? What do you think this is? That's a histological section. That's a histological image. Excellent. How did we know that these are cells, that they're not some sort of a painting? How did you know? This is something that you probably have seen in the biology class. How do we identify? Thank you so much. Exactly. We do identify tissues or cells by their nuclei. The membrane or the boundaries of the cells are not that easy to identify. So these are all nuclei. Would you tell that those are more or less the same type of cells? Yes or no? Yes, exactly. So this is a tissue of one sort. This is actually a smooth muscle tissue. Now, one more question. Do you think this has some sort of staining on it? Is it stained or unstained? Stained, exactly. Because unstained tissues are simply colorless. They have no color, and we need to stain them in order to visualize them. There is one exception where we don't need a stain. إذا حضرت محاضرة الستينينج and microscopes اللي في حالة الفيس contrast type of microscopes. لما احنا بنحاول نشوف خلايا عايشين احنا زرعناهم على petri dishes, we try to visualize them. And this is why this microscope actually was invented. لأنه stains are simply, those are toxins. These are chemicals. If we add them to living cells, it will kill them. But these cells أنا بزرعهم, بدي أكثرهم, بدي أعمل عليهم studies. I don't need them to be dead. I need them alive. فما بصبغهم, لكن at the same time I want to monitor them. So I use the phase contrast microscope. في هذا type من الميكروسكوب specific built-in opticals that allow us to see unstained ones. تمام? Now, how did we actually get this? Did we take a section from the stomach and then we sliced it? We put it on a glass slide and then we went right away, and we stained, then we went right away to a microscope and took images. Is this the correct steps to get this image? Yes or no?
No.
No. Why? Yes, elaborate. شو بدنا نعمل? What shall we do? لا توشوا شو احكوا عادي. Even if it's wrong, it's fine.
Encase them in wax.
That would be the one just before last. But excellent, yes. We shall infuse them, embed them in wax. Before that. والله انك رائعة. We need to fix them. يعني what do we need to do? We need to preserve the tissue. We need to preserve it. Now, if your father has bought some meat, shall he leave it on the counter for a couple of days till your mother remembers and she will put it in the freezer? Or she shall put it right away in the freezer? شو برأيكم؟ خليها كم من يوم حتى تعطوا بالبيت ونحطها دغري بالفريزر. Why did we put it in the freezer? Why? For what?
To preserve it and make it last
Yes,
long.
exactly. That's one way of preserving food. But we put it in the freezer because we want to eat it. But those tissues, I don't want to eat it. I want to process it. So I need to preserve it, but there's another way to preserve them via chemicals, via specific fixatives. So we need to fix the tissue, we need to preserve it, and then we need to process it. And this processing, now when we say we need to preserve the tissues, we need to add fixatives. Those fixatives will do what? هلا احنا ليش بنخاف ان التشوز بتخرب عنا؟ Why? What will happen when the cells dies or when you excise a tissue out of a living object? شو
Lose
بصير؟
its structure.
بصيرها؟
Losing the structure.
Yes, so the structure will be disrupted. لا, why? Why would this happen? That would be lie. There's something more important internally. والله انك رائعة. Exactly. Autolysis. What does that mean? اللي هم الانزيمز of the own cells will digest its own self. شو يعني؟ الخلية بتاكل حالها. So this actually it occurs within a couple of hours after this tissue has been excised. So we have to preserve it as soon as possible. You cannot leave it for a couple of hours and then you remember and fix it. If you can't do it right away, you will have to keep it in a cold surface. But طبعا bathed in specific solutions. So هاي الفixative شو بتعمل؟ لما بنحكي enzymes, what jumps into your mind? Active site مع lock and key. Now these keys, I have to lock them. How? Cross-bridging of the different antigens of the cells. That's the function of the fixative. فشو بيضل إشي للactive site? There isn't for these active sites any keys to attach to that will enable these enzymes to do the autolysis اللي بيعملوه بالخلايا. So these fixatives will preserve by creating cross bridges ما بين the different antigens اللي موجودين بالtissue. That's it, or there's something else I have to take it out of the tissue to preserve it further. مين؟ في حدا حكى كلمة صح. Thank you so much, exactly. Now I have to extract the H2O. What does our process mean, or what is the most sophisticated term that we use? Dehydration, excellent. And that's what we actually do during the processing step. So we fix it, we add the formaldehyde, which you will see in the next video, and then we extract the H2O. This is called dehydration. Now when we dehydrate tissues, or when we dehydrate, for example, if you've seen dehydrated vegetables and fruits, how do they look like? They're quite wrinkled, and they lose a lot of their shape. But how am I gonna restore this shape back again? Wax, excellent. So we will embed, we will infuse the tissue with wax. This wax will take the previously occupied space by the H2O, and then the tissue more or less will resemble as much as possible of the tissue when it was in us, and that's our aim. أنا بيهمني ضل التيشو بتشبه إلى حد كبير ما هي موجودة جواتي. And that's why those processes were designed. That's it? No. هذا الواكس كمان بدي أحطه حوالين السبيسيمن. Around the specimen. Why? Imagine هاي السبيسيمن اللي بالداخل. This is extremely tiny. يعني at best ممكن تكون 1 centimeter. We usually prefer to have it much smaller than that. You can't actually manipulate it. You can't work with it. لأنه next step, that is what? That is cutting. That is slicing of the tissue. This tiny thing we have to upload it على هالسلايسر اللي هو بنسميه microtome. So we need more wax around the tissue, and furthermore we need another carrier اللي هو عبارة عن hard plastic mold that can carry the wax with the specimen inside, and then we can load it on the microtome and we get thin sections. هاي thin sections بنحطها على البلاستيك. Got the point? ممتاز. Now, one more time. We need to fix using chemicals to cross bridge the antigens. We need to dehydrate the tissue. We need to add wax to the tissue to infuse and restore the original shape of the tissue and to have it more around the sample. And we need to cut that tissue, get these thin sections. And when we say thin sections, how much do you think it should be cut thin? Centimeters, millimeters, micrometers, nanometers? Thank you so much, exactly. Micrometers. To get actually good images, we need to get really thin sections. How thin am I talking about? It's around 7 to 9. Does that mean you cannot go higher? Yes, you can. لكن ما بتكون الإيمج quite clear. ما بتكون crisp الإيمج. Could we go thinner? It would be quite risky. And not everybody can actually get extremely thin ones. So 7 to 9, those are actually good thin sections that you can actually work with later on. Now, we've done the cutting. We have loaded the section on the glass slide. Now we do the staining. We need to cover that tissue with a mounting medium, then we put a cover slip, and that is the final product. This is how the tissue would look like. That is ready to be taken to the microscope where you can look at it and then you can identify what type of tissue you are looking at. Tamam? Now, the one you're looking at, it is stained. What type of staining? This is something that has already been uploaded. We have different types of stains. Lakin, we could easily say we have general stains and we have special stains. This is, by the way, a general stain that's called hematoxylin and eosin. And we will see lots of hematoxylin and eosin throughout this semester. And I'm sure a lot of you have probably haven't created the proper images for the processes. So we will watch the video step by step, but we will accelerate it at points and then add a few comments when needed. So we'll move to the YouTube. Why do we need the fixation of the tissue? Why is it required?
To prevent
Thank you, to preserve. Why do we need processing or dehydration?
To remove the water.
Yes, to remove the H2 and to further preserve the tissue. Mumtaz. How thin are our sections?
7 to 9
Yes,
micrometers.
yes, this one's 7 to 9 micrometers. Thank you so much. What is the most commonly used microscope?
Light.
Yes, light, bright field بنسميه bright field. الشاب اللي طلع, where are you going? You need to ask for permission. Now, when we say we need to fix the tissue, what type of molecules shall we add to the tissue in order to preserve them? Sugars? Chemicals, yes, chemicals. What type of chemicals? This is what we will be seeing quite shortly now. والله أنا أقرأ شو اسمه. Thank you so much, Abdullah. Yes. How many of you تنافس, including المكرمة? Higher. موازي? دولي؟ ما شاء الله. Okay, silence please. Silence. كميل, اهدى يا كميل.
Onto a glass slide so that we can view it down the microscope? Well, I'm going to take you through the processing techniques and the equipment that's used in a pathology laboratory that actually makes these glass slides.
Now, if you have noticed, she has said something, and that is pathology. What does that mean? What does that mean?
علم الأمراض.
والله ما أقرأ. Yes, exactly. So, we usually excise tissues in order to study them, or if a patient is seeking a medical help because some sort of abnormal growth of a tissue or a lump, usually they will excise part of that or excise the whole thing if you so close that this is probably a tumor, and then you send it to the histopathology lab, and then they can investigate if it is or if it is not. تمام? And usually this is something done during the OR or in the OR, in the operational rooms. If they're excising a tumor, they need to make sure that they have excised the whole thing. So بضلهم ياخذوا من margins, they send it to the pathology lab, and then to confirm that we have excised the whole thing or not. Another use for that, if someone is doing research, someone is in the lab studying, let's say, a drug, its effect on the lungs, its effect on the kidneys. بيعطوا هالانيمالز هاي the drugs, and then after the study period has finished, they will euthanize the animals, يعني بيعملوا لهم humane sacrifice, and then they excise these specific tissues that they are interested in, and then they do what you will be seeing now.
So that we take a big and all or some of the tissue is prepared for tissue.
So for this, they're just doing extra work. They're recording information regarding the sample. This is a section, or this is an excision from the liver, but she's not going to use the whole thing. We need to take a tiny piece of this big chunk of liver.
Processing. Some of the tissue is removed from the organ and it is placed into a cassette. The cassette is able to hold the tissue.
Now, why do we need to load it on this plastic thing? You can imagine having a tiny piece swimming in the solution that we will be using. It will be hard to manipulate and catch and move to other jars. So usually you put it in those plastic carriers to make the manipulation of the tissue much easier.
Place in the intention is to preserve the tissues permanently in a lifelike state as possible. Fixation should occur as soon as possible after the tissue has been removed from the body, and this usually occurs in the operating theatre, but sometimes occurs in the lab. Fixation reduces autolysis, otherwise known as tissue degradation. There are a variety of fixatives that can be used, depending on the type of tissue present and the features to be demonstrated. The routinely used fixative is an aldehyde called formalin, otherwise called formaldehyde.
What shall we do next? Processing, yes, which includes dehydration. Excellent.
Has occurred, the samples are placed into a tissue processor, which is an automated machine that pumps the chemicals in and out of the tissue specimens. This is necessary to remove the water from the tissue and replace it with the molten paraffin wax. The molten paraffin wax will eventually set hard so that a microscopic section can be prepared.
Now, we have fixed it, we have dehydrated the tissue, we have already infused it with the paraffin wax, and now we shall add more wax around the specimen and add the carrier that will be attached in here, plastic mold, that will be attached to the specimen throughout the process.
The embedding center consists of a hot and cold surface. The high temperature keeps the paraffin wax as a liquid so that it is easier to manipulate the tissue sample from the cassette to the mold. At the embedding center, the tissue sample is removed from the cassette and is placed into a mold. The embedding center consists.
Now, pay attention that she has already removed the sample. She added it to this metal mold, and now she's going to pour the hot wax coming from this nozzle.
Of a hot and cold surface. The high temperature keeps the paraffin wax as a liquid so that it is easier to manipulate the tissue sample from the cassette to the mould. At the embedding centre, the tissue sample is removed from the cassette and is placed into a mould. The mould is filled with liquid paraffin wax.
Now she has moved the specimen with the plastic mold to let it sit and solidify, because the wax was molten. We need to have it in a solid state that will allow us to section it. What shall we do next?
Slide.
Yes, cutting, slicing, sectioning.
The microtome is a bit like the deli slicer at the grocer's. This instrument has a very sharp knife that enables a very thin section of the tissue sample to be cut as thin as a piece of paper. It is very important to follow the safety guidelines and know how to use the roll bar and the safety lock on the microtome. The very thin sections that are cut.
Now what do I have in this ribbon? What's in it? What's in this ribbon? اللي بيطلع معنا.
Type of the tissue and the wax.
Exactly. So the tissue in the center with the wax around. هلا هذا الribbon, what shall we do with it? I'm gonna accelerate this.
Section to be smoothed out, so the section is nice and flat. The section can then be picked up from the water bath and placed on.
ممتاز. What is this here?
Picking up the tissue without the wax.
لا, this grayish whitish thing.
No. Water.
That's the tissue. That's the tissue. Why am I not seeing the wax? This is a warm water bath. So the wax has already melted, but the tissue does not melt. So when we load it, but again, الواكس مع نزول درجة الحرارة بيرجع بالsolid state تبعته. I don't care about the wax as long as I have the tissue there. Got the point? Now what shall I do next? Staining, thank you so much, exactly. We will do a staining. And that's the extent of it. The completion of this is the staining and microscopes, which you need to watch on your own. If you have any question, you shall ask me. How much time left, please? 2 minutes. 2 minutes? Okay. 2 minutes are not enough. So next lecture, we will do what's left of the epithelium, and then we shall see images together. Now, there are 4 students that I need to see them. كميل ومنك على اليمين 3 تعالوا لي. يعطيكم العافية.